Zheng Guo. I'm the full professor in Department of Biological and Chemical Engineering in Aarhus University, Denmark. Right now I'm the coordinator for this ACTPAC project. That my background is a chemist and also a biologist. So this is why we were able to, touch or contact different people with different background. Was able to complete this very fantastic project, ACTPAC. That yes, that's for why AU was able to as a coordinator to play the roles.
My name is Bekir Engin Eser. I am an associate professor at the Department of Biological and Chemical Engineering at Aarhus University in Denmark. And I am leading the research group Enzyme Engineering in our department. Our organization, Aarhus University, is also the leading organization of the ACTPAC project. But the role of my research group in the project is to lead the work package 3, which is about enzymatic technology for functionalization of hydrocarbons for plastic degradation.
My name is Frederick and I'm from Aarhus University, working in the enzyme engineering group, who works on producing these plastic building blocks, basically.
Our research group, Enzyme Engineering Research Group, specializes on biocatalysis. This is basically using enzymes for synthetic purposes. So enzymes are nature's catalyst molecules. So basically enzymes speed up all the reactions in our body, all biochemical reactions. Basically they sustain life. But enzymes are also amazing molecules that they can also function outside organisms. So what we aim in our research group is to use enzymes to speed up the reactions that are useful for synthetic purposes. For example, for the synthesis of like food ingredients, biofuels, commodity chemicals. Because the current technologies mostly use chemical catalysts, which sometimes can be not as environment friendly as we wish. They might need harsh conditions, toxic solvents, but enzymes can do the same conversions to produce chemicals in a much more benign way, in a more green and sustainable, under more green and sustainable conditions. So our aim is to replace these actually chemical catalysts with enzyme catalysts to reach more sustainable processes. In ACTPAC project, the main aim is a fully circular pathway to recycle polyethylene plastics, which are carbon-carbon backbone plastics, into ester backbone-based biodegradable plastics. And our role in this project is central to the project because the polyethylene, when it is degraded by chemical means, which will be done with our other partners in the project, they generate hydrocarbon molecules. These molecules are basically made up of carbon and hydrogens only. So there are not really many uses of these molecules other than using them as fuel applications, for example. But if we can add oxygen molecules onto these hydrocarbons, then we open up a lot of new opportunities where these molecules can be used. In other words, we are increasing the value of the molecule by adding oxygens onto them, and we are going to do them by using enzymes in a green and sustainable manner. We will add function to hydrocarbons by terminal oxidation, by using bacterial enzymes, and once they are functionalized by oxygen, they can be used to make ester-based biodegradable plastics. Our methodology, the technology we are going to develop, is a green and sustainable enzymatic conversion technology in order to convert hydrocarbons obtained from polyethylene waste into higher value molecules by adding oxygen molecules onto these hydrocarbons. So the key here is our methodology is benign because rather than using chemical catalysts, we are using a biological approach, enzymes, which are biodegradable, which work under mild conditions in water, so they don't generate waste as much as the chemical catalysts do. So by using enzymes, we are aiming to reach green and sustainable approach in plastic recycling and upcycling.
I think two things actually excites me quite a bit about ACTPAC. The first thing being that ACTPAC actually tackles this problem of polyethylene plastics and CC backbone plastics, which many other research groups really neglect to really tackle. And I think that's really cool. And the other thing is the combination of using biochemistry or biocatalysis and also conventional chemistry. And usually these two fields might frown upon each other a little bit, but actually combining them really can utilize the strengths of both in a really nice way. I think it's really synergistic in this way, as actually the chemist can do something that we biochemists cannot, and we biochemists can also do something that the chemist cannot. So really utilizing the strengths from both sides is really nice.